Really excited to be with you all today because we lined up with some new exciting results that we can share today. So you guys are going to see some new stuff. I put it at the end though, so you have to listen through all the other normal stuff. But I think you'll find it exciting. So yeah, I'll just start by introducing FutureHouse. So FutureHouse is a nonprofit based in San Francisco, funded primarily by Eric Schmidt. And we're about 20 people. We have a wet lab. That's what's in the background. And we're trying to basically solve big problems in automating science. And a lot of it is focused in the domain of biology. So before we talk about AI and science, I think over the last maybe 20 years or so, the kind of nature of science has been changing. So this is a plot of the number of papers submitted to arXiv as a function of month. And you can see sort of around where ChatGPT was introduced, we don't really see that much of a change in this sort of trend of increasing number of papers. There's a little bit of a dip during the pandemic, and then sort of picked up right around when ChatGPT was released. But basically, we've been seeing more and more papers now. And actually, I think just a couple of days ago, arXiv announced they hit 25,000 monthly submissions. This axis only goes up to 23,000. So you can see it's out of date 6 months later. And also the kind of papers. There's more papers being published every year. Also, the kind of papers being published is changing. So in 1980, the number of paper authors was about 1. And now the number of co-authors is reaching about 5. I didn't have data all the way up to today, but I think it's pretty close to 5 now. And so it's not only more papers, but also the complexity of the number of sort of specialists that's required to get a paper published is increasing. And so basically, from a perspective of someone trying to just do science, forget AI, you're having an increase in the sort of intellectual bottlenecks of doing science. Basically, you need to consider more and more papers every year. So the reference sections in papers is growing. The number of papers in your field is growing. The number of journals in your field is probably also growing as well. And so we see about 5 million papers per year. And I think it's now going to be 7 million for this most recent year. And there's about 250 million papers out there today. So you're seeing a pretty big percentage growth in the number of research papers. And also the cost of data is coming down as well. So I have here a number of $200 for a whole genome sequence of a human. It's about $1 per gigabyte of sequencing. I hear from pharma companies that it's actually closer to like $80 to $100 per whole genome sequencing. So we're seeing a rapid drop in the cost. Somebody's shaking their head in the back. You disagree with? Yeah. $500? All right. We can quibble on this. I talked to the VP of AI of Roche, I think it was, and he was saying they were down to $100. But interesting. All right. So maybe $500. It's fine. And then also there's less disruptive papers. So along with basically the increase in literature we have to go through, there's multiple measures of basically a decline in the productivity of science. I think this one is maybe a little more complicated. There's this paper put out by Park and Funk, and basically they tried to control for the number of papers that are coming out, and they basically looked at a few top journals and saw how often a sort of canonical citation was displaced by another one. So maybe there's something about how the ribosome works, and there's another paper that comes out that has a better description of it. How often does that displace the previous one? And we see that in biology specifically, the number of these disruptive papers is going down. I think it's hard to measure things like disruptive papers, but you can also see this in sort of productivity in the pharmaceutical industry, right? So this thing called Eroom's law, which is kind of out of date, is not as true, but still we're seeing over $1 billion per FDA-approved drug right now in research expenditures. I've heard people argue that's an R&D tax loophole, and also it's no longer holds, but anyway, there's a lot of signals, I think, that show that it's getting harder and harder to make advances in the frontier of science. And so basically, the conclusion of all of this is that modern science is hard. Okay, and so when we think about what are the next kind of big breakthroughs that are going to come out, they're really hard to even conceptualize as fitting in one paper or one equation or one sort of breakthrough, right? And so I think the kind of scale of science is reaching a point where it may not be just one group, or it may not be 1 or 2 groups. And even if it's a consortium, it may be difficult for a whole consortium to work together coherently on a problem. And so that's sort of where FutureHouse comes in, is that you're set up as trying to take some of the advances in AI and apply them to automate the processes of these intellectual bottlenecks that are growing in scientific discovery. So that's sort of the mission, right? So a lot of the work that I'll talk to you today about is different ways that we can try to accelerate discovery. So before we get too far, I think everyone talks about large language models, and they're doing programming, and they can schedule your restaurant reservations and book a plane ticket for you. So can they just do biology with no effort at all? So I mean, obviously it sounds ridiculous, but we did want to see to measure it. Like, is there some like benchmark we can look at to see the progress in biology? And so we looked at some of the existing benchmarks. So this is MMLU Pro. This is kind of one of these commonly used benchmarks to see how good language models are. Here is one of the questions from the biology category. So as of 2017, how many one-year-olds have been vaccinated against any disease? So when people say, these models are, getting 80% or superhuman performance on biology, these are the kinds of questions you're using to measure it. And I don't know about you all, but I don't come across this kind of, difficult intellectual task on a daily basis. Here's another one from the data set I just thought was interesting. It's the title of this album. This one I found very interesting. It's find the logarithm of 3 to the 2, okay? See, I feel like if you polled a group of scientists, they would disagree on what the logarithm of 3 to the 2 is, right? Could be base 2, could be base e, could be base 10. I actually, unfortunately, I didn't write down the answer in my slide notes, so I don't know what is the official answer that large language models choose for this question. And so, people have recognized that, and so the sort of like best effort model, or sorry, benchmark right now is something called Humanities Last Exam. Catchy name, can't forget it. But really, even look at the questions are still textbook questions. Like, these are questions which have a clear, correct answer, and it's like you're trying to just, solve some kind of known problem. It doesn't really get at the frontier of science. So at FutureHouse, we put together a benchmark. We put together 2 now. I'm going to talk about one we put out in June, but we put out a new one like a week ago. But what we tried to do is capture some of the things that we come across in our own work. So I have one on the right-hand side, which is cloning scenarios. This is made from like PhD students' lab notebooks, questions that they actually faced. And then we convert them into like questions that we could measure with multiple choice answers. Multiple choice answers is a really hard thing to do. I'm going to talk at the end of my talk about how to get away from multiple choice answers, but just it's so much easier to evaluate the models with multiple choice answers. So a lot of the work in these things is making sure they're good distractors, as they're called. But then we also have things like LitQA. LitQA was built to be a bunch of questions that you could only answer by finding some specific paper in the literature and reading the full text paper of that, the full text of that paper. So the idea is to get away from like the memorization. So we don't want to measure if the models have memorized all of scientific literature. We want to see if they can answer a new kind of question. And then CQA is just like just working with biological sequence data. We also did things that are multimodal. I won't really talk about those, but basically, can you read a table and not just like convert the table into text, but can you actually answer a meaningful question about the table? So we put out this benchmark, and we hired a bunch of PhD biologists. If any of you want to make some money on the side, you can also be a FutureHouse contractor to take tests and pit yourself against AI. But we hired these humans in the green bar, and you can see that the humans are just destroying the models in every single category, except for reading tables, which I find to be very interesting. But I think also like it kind of makes sense because we really couldn't have that hard of questions on reading tables. But anyway, so I think, and this is random, right? So this is the random line here. So most of these models are right around random. And I think this captures much better the experience of someone who does science with what happens when you go to chatgpt.com, right? If you go to ChatGPT and you put in your questions, a lot of times it's just like not missing or it's not getting the point. And I think this benchmark kind of captures that experience better. Whereas, if you see other benchmarks, they're all, the AI is superhuman already. I do want to mention like one interesting thing that we found in this is that, these reasoning models have been coming out now. And even though reasoning models actually help with some benchmarks, like protocol QA, which is basically lab protocols where we make a very clear mistake in the protocol such that it would be impossible to do, reasoning models get better at that, spotting those mistakes. But other ones, these cloning scenarios, which is coming from lab notebooks, reasoning models don't do any better, right? So there's really something missing from these models to accomplish these tasks. Okay, so basically we have to do some work. And so at FutureHouse, we've been trying to build AI systems. And so AI systems are things like agents that as language models connected with tools that you as a scientist might use, like Google Scholar or like benchling or molecular dynamics. And so basically what we're doing in FutureHouse is steps 1, 2, 3. We built an organization that is built around trying to build scientific agents that can use these tools. We measured human performance on these things, and we were able to exceed human performance. And now we're trying to get to like making scientific discoveries at scale. All right, so what is an agent? This is a real question here. I think we try to use the like nomenclature from reinforcement learning. If any of you know a little bit about reinforcement learning, basically you can't really describe an agent without describing the environment that it's in. So we have an environment here, and we have the agent that is using tools inside of an environment, and it communicates back and forth with this environment using natural language. And so instead of like, I have an arithmetic question here, and I answer it using a calculator rather than just trying to do the arithmetic myself. And this agent is a language model that uses these tools to answer the question. And sort of an agent is basically coupled to this environment, which is a set of tools that are specific for solving a task. This is super tiny, but basically we tried to put out a repo. We succeeded in making a code repository to define your own agents and use them in code for your own tasks. Like if you have a bunch of tools you want to use, you can combine them with a language model. Question? Yes.
Are all of the questions text-based questions?
In our like repository, no, you can use multimodal inputs. Most stuff I'm going to talk about today is mostly text, but some of it has like molecular structures, which are graphs, but usually we represent the graphs as a string, so it ends up being text in the end. So a lot of the work that we've done at FutureHouse is basically how do we go beyond just taking a language model, putting it into an environment, and praying that it works. So a lot of the work is basically how do you train these things to do better. And in the interest of time, I'm not going to get into a lot of detail about how we actually train these things, but it involves just sort of normal deep learning methods. Like you build a computational graph, you take a derivative, you propagate it through the graph. So before we get into the training thing, like if I just take an agent, one of these agents that on the previous slides you saw were doing very poorly, and I give them some tools, we see there's some gain. So basically I have here, like this is how well the models did without tools, and then I give them tools and they get a little bit better. Okay, I give them tools, they get a little bit better. Not a huge gain, right? So it's nice that if you just tell the LLM, no, you don't know how to do arithmetic, use the calculator tool, or no, you don't know how to read, you don't know how to translate from nucleotides to amino acids, you have to use the nucleotide translation tool. But then what we do is we actually take these models that can do okay, and then we basically let them try it out. And this is showing you like this little diagram showing you all the different tool calls that they make. So each color is a different tool. And then in this case, they're trying to do molecular cloning. Anybody who's done molecular cloning before? But yeah, I had to learn that quite a bit for this project. But essentially, we have a bunch of questions about molecular cloning, and then we let these agents go give it a try. And then the ones that are successful, we mark with green, and the ones that are unsuccessful, we mark in red. And then we throw away the red ones. And then we just train on these successful trajectories. And at the end, you basically can start doing these cycles of let the agents go try it out, see if they make the right plasmid or get the right insertion. And then if they got it right, we like take those successful trajectories and then we train the model to be like, this is good job. And over time, this is just like GPU hours. And this is like what a frontier model is. Like, if you take these frontier models, we get better and better. And these are our own models. These are 14 billion parameter models. Sounds like a lot, but actually you can run that on your laptop, no problem. And so we can get past these frontier models, which, as you know, are like hundreds of billions of parameters, and you can't run them on your own laptop. They have to be run on special data center GPU servers. And we did this on these two tasks. One of these is CQA. This is the one about molecular cloning. LitQA is about using tools like Google Scholar, Citation Graph, basically to answer questions about the literature. What's interesting is that when you start, this is like what the start looks like. These are the trajectories that we just get from randomly trying it out. And then over time, it looks like this. And so basically we start here and it's exploring a whole bunch. Sometimes it's calling like the annotate plasmid tool like 5 times, and it doesn't really help to annotate it a bunch of times. And then here it gets much more focused and then it does a better job at the tasks. It's actually an 8 billion parameter model. It's 14, it's 8 billion parameter model. Yeah. Great question. We basically have some, so the question was, how do you define successful trajectories? The tasks are things like, I don't know, what's the longest open reading frame in this plasmid? Okay, and then, so we know like what is the longest open reading frame in the plasmid from like doing it by hand. We can see if the model can figure that out from looking at it. That's example question. Like, I think they're more like how do you clone M. scarlet into this plasmid so it'll go into E. coli and express. And then it has to go like to add gene and find the right sequences and download them and then come up with a protocol will then result in expression. So in the end, we get these agents out that can run to do these molecular cloning things, and they're costing a very small amount of money. So these are basically our models here, and then these are the frontier models. So we're like 2 or 3 orders of magnitude lower in cost. One of the cool things you can do with that is that if it's this cheap, you can actually just run it 1,000 times and then take the consensus. You can just have a majority vote. So you have 1,000 agents, and they all try like, what's the next step? And they vote. Like, I think we should do Gibson assembly. And then some like, no, I don't think we should use Gibson assembly. I think we should do restriction enzyme cloning. And they take the vote, and then they go with whatever is voted for. And that actually boosts accuracy by quite a bit. So when we're done with this, we're done with training. We have a trained agent. What we do is we connect it with the environment, and we package the whole thing up, and we call it a crow. And so it's a crow because crows, the birds, can actually like speak English. You guys know this. You can go to YouTube. You can see videos of crows speaking. They can speak like a parrot can speak, but they know how to use tools. And so this is like the bird part, and this is the tool use part. All right. So here are some of the crows which we've launched at FutureHouse. Well, I shouldn't say that we've launched. So ChemCrow is something that's coming soon, made by Manu here. So if you have any questions about ChemCrow, you can talk to Manu. It's a really exciting tool for basically just doing protein design in this kind of environment. And then we have molecular cloning crow. It wasn't actually cool enough that we called it molecular cloning crow because I don't know, we just didn't find molecular cloning that exciting, so we didn't really push too hard on it. Then we have ChemCrow, which Sam, who's going to give the next talk, who maybe is here soon, she did the work on ChemCrow. And then PaperQA, which we've retrospectively called WikiCrow, which does literature research. Oh, so I do have a couple slides on ProteinCrow. I'm going to go through this super quick. But basically, ProteinCrow has some existing deep learning models, existing tools like bioinformatics. It can call our literature research agent. And then here's an example of like design 92 binders for PD-L1. So we have a video here. Yes, so here is like ProteinCrow calling the tools. So this is sort of the steps that the agent has taken here. Sometimes the models will emit like a rationale for why they made the decision. So this is like what it's thinking it's doing in the trajectory. And then it's calling these different tools that call these underlying deep learning models like protein MPNN or RF diffusion. And you can see it's basically reading through the output from these tools. And then over time, it's gathering into like a library of known binders that we want, or sorry, binders it thinks are good. And at the end, because we're, oh, did I lose my internet connection? Well, anyway, there should be like a video of a protein rotating here. But anyway, we did do wet lab validation and found that some of these binders actually did bind to PD-L1. So it's very exciting.
Sorry, I have a question.
Yes.
I'm actually just curious why you didn't sort of pursue the molecular cloning tool a bit more, because I mean, people are doing a lot on protein design and chemical design. And to me, actually, I think the most interesting thing in the molecular cloning, if you were to make it a tool, is it's one of these common lab techniques that, I mean, it so often fails. I'm sure everyone who's done cloning has an example of something that, looked like it was going to totally work and then it didn't. And I'm sort of interested if there were any conversations you and people on the team had about having a model that actually gave you like success rates of cloning and speeding that up. I think that's sort of actually the most interesting thing to pursue in that space. A lot of people know how to do the cloning, but predicting whether it's going to work, I don't know if there's a lot of tools out there that really tell us that.
So there is yeah, basically the reason we didn't really pursue it is we have to make this kind of calculus of like how impactful is the work, like where are people going to be really excited about it? And I think molecular cloning is a great thing for a lot of molecular biologists. They would be excited about it, but I think it's just harder to make it understandable as high impact work. So I'm happy to go back to it, but it's like one of these things too where you need somebody on the team to be really excited about a project to push it forward, and everybody in the biology team is just like not super excited about doing a lot of molecular cloning to validate these models. But if anybody, if anybody has the passion for cloning Crow, email me and we will happily send you designs, and you will know that your cloning experiments will be leading to a greater cause. Okay, so I want to talk now about PaperQA. So this is, again, an agent in an environment. And here I'm going to show you basically a complete sort of measure against humans, beat humans, and what can you actually do with these sort of automated scientific agents. So PaperQA has a few tools. One of them is citation traversal. So basically, I have a paper I think is relevant. Who cited this paper and what does this paper cite? Paper search, which is like a Google Scholar query. Gather evidence, which is where like, okay, I have a bunch of papers I think are relevant. Let me read them page by page and see if there's any passages in that paper that could be helpful for answering my question. And then it generates an answer, basically. It takes all the information it knows and tries to answer a question. And I probably should have said this, but the input to PaperQA is a question. Like, has anybody studied this before? Or list 3 hypotheses for how to make a room temperature superconductor. And in fact, actually now, this is out of date, is that we now actually have clinical trials as a way it can search. So it can search through clinical trials. And we also have like open targets. So it can look at things like GWAS, VWAS, human genetic data that's put together into a nice API by open targets, which has depth map as well, which came from the broad. So here's an example of what this looks like. So we have here, like, has anyone used LLM agents to automate the discovery of a novel molecular chromophore? That's the question I asked it. You can see basically the decisions the agent makes at the top left. So it's finding papers. These are some papers it's found and it's trying to download them from various sources, like preprints from open access literature. And then now basically it's reading page by page each of the documents that it downloaded. And it's basically decided, okay, these were interesting passages, so hold on to them. And then so now it's giving the answer to the question. Okay, and then yes, somebody's done it. In fact, it brought up a paper called ChemCrow, which Sam Cox will be presenting on the next one. Okay, so when we started this project, this is January 1, 2024. We have a benchmark LitQA, which I've described before. We were getting like 35% with our agent. And then over time, doing a lot of changes, improvements, making the tools better, like getting our ability to get more papers, we were able to get to basically right around where human level performance is. And we hired like biologists, like PhD level biologists to do this. And then after that, we were able to do this training procedure on it, and we brought it from 70% up to 90%. So basically, if you ask a PhD biologist, we pay them like $10 per correct question, like answer this question. You have unlimited time. You can use any tools you would like. You can use Google Scholar, et cetera. They get a 70% accuracy or so, and we get 90% accuracy on this. So yeah, pretty cool. I'm sure if we charged $1,000, like if we gave them $1,000 to get the question correct, I'm sure the accuracy would go up. But I think it's like reaching the point where it's like, okay, for $10, like I'll put in this amount of effort, and we're able to beat that effort. Now, answering like these LitQA questions, which are multiple choice questions that are on niche topics, that is not really representative of doing science. So we decided to try to find like a task that we didn't train on that is like still maybe better representation of synthesizing knowledge. And so what we did is we basically had our system write Wikipedia articles, and then we took human Wikipedia, and just to set a control, we looked at the human proteome. So we have every single gene that encodes for a protein, and we basically found which ones have Wikipedia articles written by humans, and then we wrote corresponding ones written by our system. And we extracted fragments, so like a paragraph or a couple sentences from each Wikipedia, either the AI-generated Wikipedia or the human Wikipedia. We had PhD biologists, again, grade which ones they thought were more accurate. And we beat the human Wikipedia on accuracy. And then we did another project, which I think Sam, are you going to talk about this? Yeah, okay, great. So I won't get into this too much, but one of the cool things you can do with this is you can actually take a claim, like, I don't know, the peak intensity of light of the sun is green. And then you can go see if anybody's contradicted that in literature before. And by the way, it is green. It's crazy. I don't know if any of you have little kids, but like my son is like, what color is the sun? And it's like, oh, it's yellow. And it's like, okay, but I heard at school that it's white because the astronauts on the International Space Station take a picture and it's white. And then I'm like trying to figure it out. Apparently it's also green because if you like, it's black body radiation, but the peak intensity is that green color. So I don't know what color the sun is anymore. So, yes. Question.
I just wanted to follow up on that. I actually had this question on the archive slide. Like,
yeah,
if you're training the models on the existing corpus of the science papers, what's your opinion on the reproducibility crisis? like, is there any way to mitigate it? Because if
Mm.
there are like thousands of papers, especially on archive where they're like not peer reviewed, significant, like maybe like 10, in the worst case, 20% of those are like just not good.
Yeah, this is a great question. So I'll give 5 answers to this question because I think about it a lot. Okay, answer number 1 is that we use something called the Finnish Publication Forum's ranking of journal tiers. So these are things that identify if a journal is predatory or if it's like a peer-reviewed journal. So we use that on all sources that come into our system. Obviously, it won't catch archive, but that is like one big issue. Is there's a lot of journals, there's a lot of like papers in like MDPI or something, or even like PLOS. There's like a lot of papers that are just like not good quality. So we try to use that. We also use citation count. I'm sorry, and I hope we come up with a better idea at some point in the future. But for now, citation count is a good measure of if something is like just not really. I don't want to say it's just not in mainstream. So it's like there's a lot of paper. You guys may not know this, but if you go search like, has anybody shown that like putting a magnet around your neck is going to make you like cure your cancer? There are papers written about that. There are papers about the cosmic memory of water. There's papers about all kinds of crap. It's crazy, but there are these journals, these like bottom tier journals that just publish anything. And so you have to have some kind of system, and citation count is one signal there. But I've written on my blog, plugging my blog here, I have a blog, diffuse.one, where I put my unserious academic work. So I made some tools and I've had some thoughts about how to spot fraudulent papers, but I'm not an expert in this topic. So I just have some hobbyist ideas, but I don't think we've solved it. And it is something that's even more important for LLMs because LLMs are extremely gullible. Question. Yeah, so the question is it aware of retractions and corrections? Yes, we do use Retraction Watch's DB, which is hosted on Crossref, to check if something is retracted. I will say that retractions are very like it's a volunteer run database. It's not always up to date. And so that is not really well done, but we do use the existing sources there. Question.
Yeah, so I know one major challenge for those large language models is the integration of new information. And in research, I think this is like a super important thing since the new development in the last 2 years would be very fundamental for people to solve the new biological question. So how do you plan to tackle this?
Yeah, so this system that we're talking about, this agent system, we tell it to only use information from the sources we provide to it. And so we've really tried to make sure that the model is only using scientific literature or the clinical trials or the human genetic data, is only trying to use that to answer the questions. And one of the reasons why, I don't know if I should mention this, but like every time it emits a sentence, every single sentence has a citation, and that citation is to a page number on a source. So you can track the provenance of everything that comes out of this model down to like the individual page in a research paper.
Sorry, I think what I mean is like, like if you want to build like a AI biologist, how do you make sure that how do you feed the new information to it without retraining the big model?
Right. So I guess what I'm trying to articulate is that this model tries to only reason from information we present to it. And so we just make sure the information we present is from like recent papers or from sources that we're confident in. And so we're trying to, I mean, you're getting at a good point, is that like the dream is that you can separate out the model's knowledge corpus from its ability to reason. Because right now they're somehow coupled. So you need really, really big models to do reasoning, but then you like have all these problems of like, then you need a really big model. And if it's like 3 years out of date, it doesn't know more recent stuff. And so, yeah, we try as much as we can to separate out like the ability for the models to use information and reason from their ability to memorize all of science.
Related question.
Yeah.
Is it possible to validate this notion that like whenever it is telling you something, it has the citation that's directly from the thing,
Yes.
et cetera, et cetera?
Yes. So in fact, this is like the majority of this eval here was to make sure that the citations and the claims that are in the Wikipedia articles it writes are consistent with the site sources that it cited. And so this like 86%, those 14% like failure, that is when it cited a source, but it either mischaracterized the source or it made a claim that didn't cite a source and it should have been cited. So those are examples of failure. So that's the sort of failure mode of that. And actually, that's how we eval. This whole eval is just, are you consistent with the sources you cite and are you accurate within the context of those sources? So humans hallucinate at a higher rate than the language model. Wait, one more question, and then I have so many more exciting things I have to talk about.
That you train the model from scratch, or do you use a pre-trained LLM and then tell it to only look at these sources? Because if it's the latter, it may be difficult to tell if it's indeed only looking at those sources or using.
This is a good question. So this plot I'm showing here, this LitQA benchmark, we made LitQA two different ways. We made LitQA, like we made a set of 60 questions or 55 questions or something, where all of the papers were published after the cutoff date of the pre-trained language model. So those papers were not in the pre-trained corpus. And then what we did is we asked it questions that it had to look at the paper to answer. Okay, so then we're like measuring, does it really like look at these papers, or is it just trying to recall and then like attribute post hoc? And the second thing is that we also check on the data set, like we go to these pre-trained models and we ask the questions of the pre-trained model without access to these tools, without access to the papers, what's the answer to these questions? So let's have a measure of like a baseline performance, and I think it's like 25% on this benchmark of how good the pre-trained model does without access to the tools. And then to the other point is that when this bump from like here to here is when we switched to our own model that was 8 billion parameters. And of course, 8 billion is a lot, but that's not enough to fit all of scientific literature. So it's not really at the same scale of something like GPT-4, which is over 100 billion parameters. Awesome, great questions. I just want to show you guys like one quick application here. So like here's an example of a question that you can ask of this. Is there genetic evidence supporting TREM2 association with tau-mediated neurodegeneration? Speculate on the plausible mechanism in your response. Okay, and then it gives some ideas about this, and it cites these, and this is about the sources, say like on page 1, page 8 to 9. So you can trace back to see where it came from. And then if you look at the sources here, you can see which sources it came from. And then you can see all the reasoning process, like why it chose different sources, what score it gave the sources, how it came to the conclusion of them, et cetera, et cetera. Cool. So I have one more, which I'll show here, is we have this website, hasanyone.com. And you can ask a question like, okay, has anyone ever made split GFP? That's not too easy. Okay, let's see. Has anyone determined the specific mechanism by which PFNA disrupts thyroid function? So we can click over to this one, and then it will go through and basically give you an answer. Like, no one has determined this conclusion. And it has some description of why it came to that conclusion, and it has these sources cited. And then you can go back and you can see, okay, like what did it cite? It cited this paper, which has 5 citations. It's from a peer-reviewed journal. This one has 81 citations from a peer-reviewed journal. This is 250 and from the highest quality peer-reviewed journal, et cetera, et cetera. And you can see like how long it thought for. This was a cached question, but it took 3 minutes to answer this question. Cost 32 cents. I don't know why you guys need to know that, but that's how much we paid. Yeah. Cool. So then the last sort of application of PaperQA I'll mention is this WikiCrow. So there's a website, wikicrow.ai. And what we did is we basically finished off Wikipedia for the human proteome. So there's about 2,700 articles on Wikipedia for the human proteome. So there's quite a few missing. There's about 20,000 protein coding genes in the human genome. And so we basically, finished writing about 17,000 articles. Took about 48 hours. And so then you have like an up-to-date version. And I'll mention too that some of Wikipedia is out of date. We can run this every 48 hours, to update the knowledge of science on the human proteome. So if you go to the website, you can put in any random gene that you want, like LIPG, and then it has some information here about the gene and the structure, and then it has the overview here, and this is all generated from our system. And you can read through it, and basically, if you ever come across a gene from some kind of screening and you don't know what it does, you could try this out, and maybe you find it's interesting. One of the cool things about this work, though, do I have this slide? Yeah, okay, great. So one of the cool things about this is that there has not really been a text embedding of the human proteome, right? So like you can obviously embed the proteins, or you can embed the genes, but what we're able to do is actually, since we have now a text description of what is the function, what's the disease interactions, like what are the physical interactions of all these different protein coding genes, we can actually embed them all. So like, take all the words in it and then find out which ones are similar. We have a complete embedding of the human proteome, and you see like things that you expect, like here's a bunch of receptors, right? Here's a bunch of DNA-associated ones. And inside the DNA-associated ones are a lot of transcription factors. And so what we did from that is we actually took that embedding data and we built this related genes section. So these related genes are genes that have like a close text embedding that you wouldn't like find from any other way except for synthesizing the knowledge of the genes. Anyway, kind of cool, maybe interesting.
So if you embed the entire text summary of the gene, I imagine you get something very different from taking each section or subsections and embedding those. And you get a much more diverse neighborhood trying to find, okay, these set of genes are membrane proteins in these types of cells versus the entire more averaged out thing. Is that an experiment you ran?
So actually, I think these are run on just the overview.
Okay.
Because what you're getting at is important, because for some genes, we don't know what like disease associations they have, so that section is empty. So this is run the overview, which is like a summary of all the sections. But you're right, you could do something like, I only want to know the physical interactions, and then you can build like a new physical interaction graph that is up to date, and covers as many papers as we possibly can. But it might be more interesting as a tool for then probing like what kind of gene or which protein-protein interactions should I be looking at? Yeah, cool. So, like I said, one of the things that's unique about FutureHouse is we're like an engineering group and a science group. So rather than just make this like a demo thing, we like built it into a platform. And so we can run, I think these are out-of-date numbers. Basically, we can run 25 things per minute. We have about 150 million papers we can look at. And so we could write a Wikipedia page for every human disease every 3.5 days. So basically, every 3.5 days, we summarize all the new literature, rewrite it for all the 25,000 human diseases. We could also check for contradictions in every single paper on arXiv that comes out every month. Basically, summary of where this fits. Does it disagree, agree? What's the consensus on literature? We're getting there, but we're almost 10xing this capacity. And so we could check every single paper that's published every year for contradictions with literature, disagreement literature. We could rewrite all of Wikipedia every 3 weeks as well. Cool. So now that I have exhausted almost all of my time, I'm going to tell you about exciting new results. Okay. So over the last couple months, there's been a real big change in how language models work. So we have been stuck on this paradigm called pre-training for a long time. This is probably something many of you are familiar with. It's basically to get a smarter model, you need more data, you need more compute, and they got to get bigger. This is like GPT-2, 3, 4, right? GPT-5. And it's become a really difficult thing to scale. And it also is a very interesting phenomenon is that pooling all of your compute into one big model and then serving it for a very tiny cost to you is a great business strategy. And that means is collectivization. Basically, all of the compute is being concentrated in just a few organizations because they can make these really big models that then have a low marginal cost. And then there's this thing called scaffolding. This is mostly of the work that I presented, was that we basically use domain knowledge to put together some tools and information that help the language models do better in scientific environments. There's a new kind of paradigm which has come out recently, which is reasoning models. And so reasoning models require domain knowledge, but they require much less data and they require much less compute. And so I think we're going to see like a reverse of the trend where it's basically becoming more and more out of reach for academics to do any kind of work in this area because big models just win. And so now things are actually flattening out where you can actually have small models that then you can use at a more expensive compute. Like it takes 10 minutes for them to answer a question rather than 10 seconds, but you can get the same performance and we can make them domain specialized. So I'm going to skip over this, but basically I'll just want to show you some evidence of this new dimension of scaling. So this is a benchmark, humanities last exam again. So basically here's this sort of typical scaling. We have like GPT-4o, pretty big model, then Sonnet comes out, it's a little bit bigger model, and then O1 comes out. And then we have this like change from traditional pre-training models to now reasoning models. And you can see this benchmark went from being whatever, 8% to then becoming like 30% in just a matter of a few weeks as models were released that were reasoning. And you have this like new dimension where you can basically make the models not just bigger, but you can make them think for longer. You can make them specialized in domain by training them on reasoning in one topic. And this has exploded into the open source community. So here's a plot of a bunch of recent models from mostly open source groups. And this is model size. And then this is the accuracy on mathematics. And you can see that we are getting models. This model here is 1.5 billion parameters. It is matching V3, which is 680 billion parameters. So this model costs probably $100 million to train. This model costs about $500 to train. And so we're able to get this huge flattening of these language models with reasoning. And you can see everything at the top has reasoning. And so reasoning is just completely, just destroyed our predictive power in this field. These Quen models are like, as you increase the model size, the performance gets better, right? And even it starts to diminish in returns as you make them bigger and bigger. These are all trained in the same pattern, completely gone. So now we have like a real big, disruption in how we think about language models. And so the question, for us at FutureHouse, and I think many people in the community, is can we do this in scientific reasoning? Because it's only been shown for mathematics yet, but there's now this opportunity to try to apply this kind of new economics of language models in scientific domains. So here is some output from a reasoning model we've been training on chemistry. So what happens is that you still have these sort of like chemistry specialized models where you can give it molecules and predict a property of it, or give it a reaction and predict the reaction outcome. But now it gives you a reasoning trace about how it came to the conclusion. And so what happens is we're building these domain-specific models that have these scaling properties by reasoning in a very long thought in English. So this is one where it's just, this is kind of a simple chemical reaction, but you can read through like how it reasons through the reaction. I think it's very exciting as a scientist to be like, oh yeah, I can see how it came to the conclusion. But also these reasoning tokens improve performance. I'm going to skip over this plot here, but basically the blue one is our reasoning model. This is very early. This is like literally trained for like 3 days. So it's going to need to be cooking for a little bit longer. But the red is O3 mini high, which is sort of the best frontier model out there. And so we're beating it in a lot of these different tasks. I'll show you guys some examples of the tasks in a moment. We have human comparison for one of these things. And so we beat humans on predicting the outcome of a chemical reaction. It's very exciting time for us, FutureHouse right now. So what the idea behind reasoning models is that we don't actually have to have a bunch of like prompt completion, like a bunch of work through problems. We need instead is verifiable rewards. So here's an example of a verifiable reward. Propose a modification to this molecule to increase its solubility by about 1 log S. It's about like, log S is the solubility in water, without affecting its scaffold. So this is kind of like a question that a medicinal chemist might ask. Is like, okay, I need to like adjust the solubility of this molecule, but I know it has drug activity, so I don't want to adjust the scaffold. So this is an example of a question where there is no one right answer, but we can verify a proposed answer. And what's exciting about this is this kind of represents a lot of the tasks we do in biology or a lot of tasks we do in chemistry. Is like, not often there's like one solution, but we might be able to characterize a proposed solution. And it's also possible of doing new tasks that was just like never possible before. So here's an example of a task which is totally insane. It shouldn't work. Okay, I'll read you the question. The reasoning is kind of detailed, but it says a C17H15O7 metabolite from Lathyrus linifolius was identified. What's a biologically plausible compound for this as smiles? So this is the molecular formula. You can get molecular formulas dirt cheap. You get these from mass spec. There's probably millions of these formulas of things extracted from organisms. Finding the molecular structure is actually very hard and requires like an expert to look at NMR data. It takes a long time. Here, the model reasons through like knowing about the metabolism, knowing this kind of organism class. What are possible structures that could fit this formula? And we gave this task to humans, and they were just like, don't be silly. No one can do this, right? Basically, it's really hard as a human to sit there and reason about all this while building up a molecule that has the exact stoichiometry that what you want. And this is the reason it's very interesting to read through it. And basically, it comes to the conclusion about what is the right structure. And this is one of these tasks where when you see the training curve, if the model gets 0%, 0%, 0% after like whatever, 12 hours, no frontier model gets anything non-zero, then all of a sudden it starts to be able to do this task. It's super exciting. Question? I can just repeat your question if it's short. Oh, there you go.
I've worked with people before who have actually been trying to use, who have found it difficult to get actually, I mean, maybe this is last year, maybe the world is different now.
Yeah.
Have found it difficult to get LLMs to produce smiles that are actually like syntactically correct.
Yes. So let me show you this right here. This is, can you make a molecule where I've completed half of it? Okay, so I have a molecule, I've completed some fraction of it. Can you complete it and make it sure it's valid? Okay, so it gets 96% on this task. In the frontier model, O3 mini gets 0% on this task. And so you're right. Now, to be fair, like if you do O3 mini on like a small molecule or like something simple, like what's the smiles of arginine? No problem. But these are all very large like natural products, and they're very hard to close all the rings and satisfy all the valencies.
So is this something where this is like a one of these submodules that you were talking about before, where you can call a?
No, this is it's just a one language model, one purpose-built reasoning language model only for these tasks. So no tools involved here. Very exciting. And it's also small enough that it can run on a laptop. So I think when we get this model finished, people can fine tune it on whatever chemistry domain tasks they want. And I think we're going to set up a recipe that people can repeat for, protein function, for enzyme function, for genome models. Awesome. So I think this is, an example of like just one of the things that's been, I think, one more slide. Yeah. So one of the things that's been difficult. Are we out of time? Oh, okay.
Oh, sorry. One more question. I think what might be interesting with this and really show its power is sort of like zero shot is kind of cool. So like with Evo, right? And they have the cast system.
Yeah.
Do you know how many samples they drew?
I think they drew 1,800.
2 million.
They drew 2 million casts.
Yeah, to do the in vivo validation, right? So they drew 2 million of them.
Oh, they sampled 2 million.
Yeah, and then
yes, yeah.
and then they did
yeah,
alignment and all these things.
Yeah,
so
yeah.
Then they got 11, but then only one worked.
Yeah.
Because they have 11, you know that they didn't initially pick 11. They probably picked 10. The 10 didn't work, and they picked another one because no one picks 11. So I think, I think one, and it was called zero shot, but whatever. So I think this is kind of cool if you could demonstrate chemistry just to match them and show no, it's like truly zero shot. I know, sorry, it's a bad question. It's but I think that's kind of cool actually. If you can do truly zero shot.
Yeah, so I think you're onto something there. But I do want to say is that if I'm given 2 models, one model is really good accuracy, and one model gives me a long page reasoning description in English about how it came to the conclusion, I would rather have that one. And it really is very interesting to see the conclusion. What's really nice about the reasoning is you can see where it makes mistakes, right? Or maybe it doesn't get it right, and you can say, go back, but assume this, right? I think the reasoning itself, is just so powerful as a scientist, because these models that are put out, like Evo 2 is great. And I think there's been some great chemistry models out there. There's some good, protein structure prediction models. But imagine if you went to AlphaFold and you asked it to fold something, and then it gave a bunch of reasoning about why this fold goes here, why this fold goes here, right? Or why it thinks this should be helix. That would be monumental, right? And I think that's one of the missing pieces for all of this stuff. Is like, okay, great, you can do black box accuracy, but explain why. And so I think this model, even if maybe it's not as good as the best model in some of these tasks, maybe there's probably a better model that's out there for some of these things. The fact that it reasons in English and that it can do all the tasks, right? You can combine 15 different tasks in here. Make sure the solubility is here. Make sure it's not carcinogenic. Make sure it's commercially synthesizable in one step. It's really exciting stuff. All right. I will just mention that we actually gave the reasoning traces to medicinal chemists that are in the pharmaceutical industry. We don't know how to evaluate these things, so we did way too many, too big of a rubric. But we checked to see, is the reasoning relevant to the question? Does it go from the prompt to the end? Is it faithful? Are there any reasoning jumps, or it just ends halfway, or doesn't go all the way to the end? How much is it like a human reasoning? Is it making any mistakes? Does it hallucinate a structure? Is it propose something that's not the right name of a structure? Does it explain it? I actually don't know the difference between this and humanness, but I'm not sure. And then readability is are all the smiles valid, or things like that. So again, we don't have a baseline. We're figuring that out. But the medicinal chemists were all very impressed and thought it was a really interesting, useful tool to be able to read the reasoning in these predictions. Awesome. So we will release the model weights soon and the technical report. But I think it's just such an exciting breakthrough. And I think it's just going to, this topic of being able to build these models, we're talking 50,000 verifiable rewards. So I think in the past, it was okay, you need to bring me 50 billion tokens for me to train a model in your domain. Now you need to bring me 50,000 verifiable rewards, which I think is very achievable by many different groups. Awesome. Yada, yada, yada. I will just mention the last thing is we actually, I think, have made a lot of really great scientific agents like cloning crow, like protein crow, like PaperQA. We're going to be releasing a platform soon where you can go, and by soon I mean within weeks, where you can go and use these things yourself, and it will have an API. So if you want to use these things as tools in your own workflows, you can call the API, or you can use them. And we have a super cool design. Design is so cool. It's like driving a spaceship. I don't have any pictures of it, but it's going to be great. Cool. So obviously, huge team here. Sam Cox is going to give a talk next about some of this work. Mike Skrilinski led a lot of the PaperQA engineering work. And then Manu. God, I hope you're still on here, Manu. Oh, no. Were you on here ever? OK, we got to get Manu's picture. Manu did the work on ProteinCrow. And then Sam is the director of FutureHouse. And yeah, we have a big team contributing to this. And with that, I'll take any additional questions. And thanks for your attention.
Thank you. So yeah, I'm Sam. I'm at FutureHouse and also at University of Rochester, and I'm going to be talking about work that happened in both of those places on automating chemistry workflows with LLM-based agents. I'll start with ChemCrow. This work is kind of old now, so I know that all of you have read it and studied it very deeply. But this is kind of silly, but when I was starting this project, I was like, I don't really know how good LLMs are at these things. I don't have a good intuition for this, but LLMs probably do because they're very self-aware. So I asked, what are you good at and what are you bad at when it comes to science? So they're very good at doing like basic fact retrieval, very simple problem solving, and they have a high level understanding. But they're really bad at like obviously any updates. So if you do any computational work or if you need to reference any literature, it doesn't have updated information. It's really bad at advanced problem solving, and also it tends to make errors very confidently. So when we're thinking about like trying to implement these, into chemistry workflows, we started thinking about agents. So ChemCrow follows a chain of thought process where you give it a bunch of tools and prompts. The tools are just like Python functions, like convert selfies to smiles, or convert the name, or look up the price, plan the synthesis. And it thinks, okay, what do I need to do? What have I done? Where am I at in this process? Which of my tools is best suited for this subtask that I need to do? What should my input be? And then it uses the tool and decides and tries to figure out what the tool told me and where do I go from here. So it's able to iterate and like react to failures and do like a react process. When we were developing ChemCrow, the way that we thought about this is we really wanted to kind of make it an assistant where it like completes chemistry tasks. So we thought, what kind of questions do chemists even want to answer? What tools are needed to answer these questions? And then where does the agent fall short? Because when you're developing an agent, it's only as good as the tools that you give it, and it can only do what the tools that you give it. And so we ended up giving all of these tools here, where we have search tools like PaperQA, like Andrew mentioned, web search, a bunch of conversion molecular tools, synthesis tools, safety, and then some basic link chain tools like, man, I don't know, let me ask the human, or let me try to run some Python code. The thing that's really difficult that we all probably know, and Andrew also mentioned, is how in the world do you even evaluate these things? The first method, which was really popular at the time, is just asking GPT-4, hey, you're in the role of a chemistry teacher evaluating two students. One is GPT-4 and one is ChemCrow. Your task is to basically give them like a review, highlight the strengths and weaknesses, and then also give it some grade. This is a really unbiased method. All teachers are very unbiased grading students, but we found that GPT-4 can't evaluate the responses that it can't answer. So if you ask it a really hard question and it gives you a nonsense answer, it doesn't have the knowledge, obviously, to evaluate it. And so it ends up grading based on, it like tells you that it's really good chemistry, but actually it just thinks that it like flows very nicely, or the grammar is very nice, and it evaluates kind of nonsense. So we needed another type of evaluation, and we used humans. So we gave a team of chemists and chemical engineers the question, and then both final answers formatted identically. And so what we're seeing here is on the left, we have the delta mean expert scores for GPT-4 and ChemCrow. And in each category, we have increasing difficulty going down. So for organic synthesis task 13 is much more difficult than task 7, where difficulty is like the complexity of the task, the number of steps required. And we see that like GPT-4 is not bad. It's able to complete some of the tasks. Humans actually prefer it on like 14 and 4 and 2 and 10. But as the tasks get more complex, it really drops off. We see that in both organic synthesis and chemical logic and knowledge. This is not surprising. I don't know if you've ever tried to ask like GPT-4 to plan and execute a synthesis. It can't do that, so this is not super surprising. And so we found that GPT-4 gives complete responses, which is great, but it has a lot of hallucinations that are very believable in some cases. It's hard to interpret, so it doesn't tell you like how it thought about it necessarily. It just gives you an answer. And it doesn't have access to any up-to-date information. So some of these would require like reading a specific paper or running like a Python package that it just wasn't able to do. Whereas with ChemCrow, we found chemically accurate solutions. It's great because it's modular and extensible, so all agents. So if you provide like you decided, hey, I really want to do protein design, you can provide it with some protein design tools to do that. Although chemistry is easier. And then it still occasionally has flawed conclusions because it's limited by the LLM, right? Like it still is going to make mistakes. And it's very limited by the tool's quality. But one of the coolest things we did with ChemCrow is we gave some chromophore data and we said, gave it some instructions all the way on the top left, clean the data, use only the data with a specific solvent. So filter it, process it, train a random forest machine learning model, make predictions, and then suggest a synthetic plan for one with the wavelength closest to 369 nanometers. It did everything that we said, and we get a final answer. And then humans actually synthesized the proposed molecule, and we had it sitting in the lab, and it followed all of the instructions. So we were actually able to synthesize like a novel chromophore with ChemCrow, where the only human intervention is giving it the data, telling it to do it, and then testing it in the lab. So with ChemCrow, we found that LLMs can use tools to complete chemistry tasks, which is not, I think, like a super trivial thing. And it can also complete novel discovery and design. So we can do novel things with it, which LLMs can't do on their own. But we also found that agents are limited by tool quality. And I didn't go into this, but you can read the paper. Safety is a really important concern when you start giving LLMs the ability to do chemistry and like hook it up to a lab, obviously. So that's a big consideration. So the next thing we started thinking about is like, okay, cheminformatics is cool and super difficult, but what if we wanted to do like a really complex task that maybe runs for a really long time or has to handle tons of files or has to do interact with like multiple packages that are very heavy? And so we started thinking about automating molecular dynamics workflows with LLMs also. So the way that this works similarly is it follows like this React framework where it thinks, acts, observes, and then it decides if it needs to complete the next subtask or react and troubleshoot. With this one specifically compared to ChemCrow, that react and troubleshoot is really important because MD is pretty finicky, and so it has to react and like update its parameters and change things as it goes. This one had, or MDCrow has currently about 45 tools, including, again, PaperQA for literature search, and then using databases like UniPro and that sort of thing to get up-to-date information, downloading PDB files, and then also interfacing with the PDB files, simulating with OpenMM, using PacMol, and then also some analysis tools using MDTrash. All of the things that we implemented were in OpenMM and MDTrash because it was easy to evaluate if it's all in one package. But again, these are modular. So if you wanted to do GrowMax, you could implement it into GrowMax. But that would be more difficult. We did a series of 25 tasks ranging from 1 to 10 subtasks, where one might just be download the PDB file for protein ILYZ. And then a 10-step one also is shown here, where we have to simulate at 2 different temperatures. We have to do a bunch of analyses, and then we have to look at before and after and do some comparison. Okay. So what we found in this project, which is super interesting, is if you look all the way on the left, we have accuracy, which were just evaluated by humans if it was able to do it in a reasonable way and get like a reasonable response. If you look at the purple line, GPT-4o, like we kind of expect out of these models that GPT-4o would be pretty good, but actually Llama 3, which is an open source model, is pretty equal. Mm-hmm.
I had a question about the task the MD like initialization.
Yeah.
And I was curious, like, how does the LLM pick the parameters? And also, I don't know, because I remember from Andrew's presentation, also there was like some like logic to how it picks and decides which task to do. How, like, if there's like, how do you approach changing parameters? Like, if it like, does it do one set of parameters and then change to another set of parameters if that doesn't work? Or yeah.
Yeah, so we have like default parameters for a lot of them because just defaulted in the code, like all the software does also. But it chooses the parameters based on the prompt. So you might say like the temperature or the ensemble in the prompt. Otherwise, we kind of let it choose. one thing that we found really interesting is that if you don't specify the ensemble, it just kind of picks whatever it wants, obviously. But the models have favorites. So like Claude consistently, always, no matter what, wants to do NPT. And you kind of have to like force it to not do it, which to its downfall. And then the other models like to like diversify a little bit. So this is something that actually with Manu also we're thinking about building like a more stringent eval set for to make sure that every single like minute parameter is defined correctly.
Thank you.
You said something like, oh, it's surprising that Llama actually performed as good as the other bigger models. But then to me, okay, the task here is like pretty straightforward. Like processing, getting PDB, processing PDB is something like you can just write, you can hard code those processes. So I wonder, in that sense, maybe it's not that surprising that the smaller model works as good as the big one.
I think it's not necessarily the case that it's super simple because or like super straightforward. And actually here we test this is with GPT-4o, but we test with like just actually having it write the code, like hard code it, which is the direct LLM. And then also just having it do a React where we don't give it any tools, but it's able to write Python code. And it actually performs pretty poorly on these. So I don't think it's necessarily the case. I think that where some of these other models fail, so, I mean, GPT 3.5 never stood a chance, but the other models that are more medium, they really just fail in using the tools correctly and choosing the parameters.
Wait, so I'm trying to understand like where do they fail? So like choosing like coding that part to me is it should be like there are only this few common lines you can write for running MD simulation. So do they mostly fail at the choosing the right parameter part? And what do you mean by wrong parameter?
Yeah. So for instance, it would like forget to choose parameters. A lot of the ones that perform more poorly, like actually the medium ones, like the Claudes and the like GPT-4 Turbo and the Llama 70B. A lot of what they do is just because the process is so long, right? Like running an MD simulation. And also like if you ask it to do 15 analysis steps, it just kind of forgets things. And it will just hallucinate. So it will like, or it'll give what I meant with the wrong parameters is not how I said it, but the like wrong file. Like it'll try to run, if it runs two different simulations and then it's supposed to do RMSD on both, it'll just do it both on one. And it just mixes those things up.
Okay, interesting.
Yeah, like very silly mistakes. Yeah.
There is one question online. Can you unmute yourself and ask the question?
Oh, yes. I'm curious, what is the contribution of large language model to WikiCrow? Is it used to convert human requests into a pipeline of existing tools? And how does your tool compare to a pipeline of tools? Thank you.
Yeah, so the WikiCrow is an LLM agent. So it takes the user request and then reasons through what tools it wants to use. All of the tools, most of the tools are not LLM based. They're like just computational tools that are built on regular software. But it does the reasoning process of choosing the tools and then the inputs to the tools as well.
Got it. Thanks.
Cool. Okay, cool. So one of the other things we found, and this is some getting at what the question I was asking a second ago, is all the way on the bottom left, we have each of the models and then the accuracy as the number of subtasks required increases. And you can see like GPT-4o, even which is like one of the best models, still kind of dies off after a while and only gets, like as it gets more complicated and we ask it to do more things, it still is not 100%. And so there's still a good way to go with this. A lot of what we saw with GPT-4o is it doesn't really skip steps. It's really good about like completing the right steps, and actually the same with Llama 3 405B. It's really good at like completing the steps, say like checking them off, calling the tools. But again, it like gives the wrong parameters and it makes silly mistakes like using if it runs at 2 different temperatures, it uses only 1 temperature and just runs it twice for RMSD. Whereas like Cloud 3 Opus and even Cloud 3.5 Sonnet will give you kind of silly things where it's like, it won't run RMSD and then it'll just give you a number like it did. It's very honest that way. I kind of talked about this, but just to come back to the MDCrow built on GPT-4o versus React and just asking the LLM to write the code. Similarly, we see a trend where, again, the performance drops off, even for the MDCrow as the complexity increases. But for the React and Direct LLM, it can complete some of them. Certainly the one-step one, it can write code to fetch a PDB file, and then it drops very quickly with like no performance after that.
Sorry, just a question. If like, how would this table look if you added the recent reasoning models like DeepSeek, the O3 from OpenAI? In your opinion, would it like strongly improve or just like marginally?
I definitely think so, because I think we have not run MDCrow based on reasoning models yet. That's something that we are doing now.
Let me answer this question because actually we've been thinking about this at FutureHouse. We tried O3 Mini High, which is supposed to be the best model in like PaperQA. We tried it in the molecular cloning tasks, and it got 0% every single time. And the reason why is that it doesn't seem to handle tool descriptions very well. And I don't know if there's some like additional work they need to do at OpenAI or something, or it's just like not ready for calling tools. But it would basically always forget to submit the answer, or it would like always forget to like download something. So I think that one's still ironing out some kinks. But I think in theory, yes, reasoning models should do better. And I think we also tried Claude 3.7 thinking, and we didn't see any difference with Claude 3.6. So we haven't seen a gain there, and I don't know why. But I mean, I think that, it's kind of obvious that the next step for these things is to let them think for longer because it's such a complicated environment. But yeah, we haven't seen any gains, zero shot yet. So the question was DeepSeek R1. No, DeepSeek R1 doesn't seem to call tools very well. And one of the problems is reasoning models are very, very long generation. And then when you do an agent, you have a very long prompt because you have a bunch of tools you have to describe. And so actually run out of context quickly in these settings.
But certainly, I think, yes, not the using tools would not do well. But in this comparison, if we asked it to do the direct LLM call, it certainly would be better.
I'm trying to understand, like, okay, I understand this might be like a first test case for something that will be more complicated later. But then just in the case of MD, I'm browsing over your preprint, and then I realized that this workflow is what most MD scholars would do and have code for already. So it's a pipeline that you can basically fully hard code. So then what's the point of using large language model to recode each of those steps? Like, for example, retry PDB is literally just, wget with a PDB ID. Yeah. So what's the point of asking the LLM to recode those kind of repetitive lines?
Yeah, so I think if I understand your question correctly, the point that we were trying to get at with MDCrow is trying to see, like we did ChemCrow and we saw that it can do some chemistry tasks, but those are relatively simple. And you get the answer back immediately. There's no file handling. It's very like simple string in, string out. And then with MDCrow, we wanted to kind of test that and bring it to the extreme of seeing can LLMs do the more complex processes with the more complex tool calling.
Which part of the process is something that requires like reasoning, for example? Like for example, I think download PDB or run simulation or run analysis, those are pre-existing packages. So which part is the reasoning part coming?
So the reasoning then comes in and the choosing the parameters for the tools. So if we don't give it all of the parameters for the simulation, for example, or if it has to look up, like if we say run this at appropriate, run some simulation at appropriate temperature based on this paper and literature and doing that piece, and then also breaking down the task into subtasks and actually making sure it completes all of those. But we weren't necessarily trying to evaluate its ability to reason about MD on this project.
Got it. Thank you.
Yeah.
I'm curious if you looked at all, and I know this also was, this project was maybe a couple years ago, but if you've looked at all at memory or at storing memories. I just thought of it when we were talking about the reasoning models, kind of forgetting things that go out of the long context window.
Yeah. That's a great question. So we also implemented memory in MDCrow. We implemented it as kind of like a chat feature because like you don't want to sit there and have a chat bot that runs a simulation that you have to wait on. But we also store them. So this is like an example of how you can like chat with it. But the way that it implements memory is it takes the entire trajectory that it ran and its thought process, and then just uses an LLM to summarize that and store it into memory. The way that this is like a kind of silly example of breaking up the steps, but the way that like I ran this in practice is if it like really screwed up, I would be like, hey, remember when you did that? That was wrong. Can you try again with some like change to it? So we did implement memory in this as well. Yeah. Okay. The other thing that we tested with MDCrow is really diving into that like increasing complexity. And the way that we did this is we built another small set of 10 tasks where they like literally build on each other. Like if step 1 is just download a PDB, step 2 is download and clean it. Step 3 is download, clean, and simulate. So it's just literally building on each other. And again, these are like relatively simple tasks. But we find that some of the models like GPT-4o and Llama 3 still have a pretty good, the diagonal is perfect, where the number of subtasks completed versus the number of subtasks required is like pretty diagonal. But interestingly, we see that like all of the Claude models really are not great. And the reason for that is we tried to be very objective, and we didn't like look at the performance of one model and then adjust our system in any way. We tried to just have like, okay, we built the system, can they do the tasks? But if you were to fix it and look at the Claude models and say like, hey, they're really bad at these 4 things, they keep making the same mistakes. We didn't see this with GPT-4o or any of the other models where they like consistently all make the same mistakes. Claude just really, really likes NPT, but is really bad at it. And so if you do make some adjustments, like you kind of default the pressure to a reasonable pressure, or you make some like not great adjustments, you actually can get the performance like pretty decent and get it to match and get it to bump up there. All this to say, like even with, we see like pretty good performance with the open source model, but even like just with Claude, you can get pretty decent performance if you really wanted to use Claude by making like model specific adjustments to the system. Okay. So with ChemCrow, we found the same conclusions, and with MDCrow, we found that like even the new LLMs can't handle these like complex MD tasks. Again, the tasks are not complex to like somebody who does MD regularly, but they're complex for an LLM. And also open source LLMs are pretty comparable to the frontier models for MDCrow, which is very interesting because MDCrow still struggles with these more complex tasks, these more like multi-step, these like long trajectories. Okay. So the last thing that I'm gonna talk about is ContraCrow, which Andrew, I think, kind of hinted at. And this is using PaperQA to detect contradictions in biology literature. So this is thinking about how we can use these LLM agents to a scale that like humans can't do. Because like you said, humans can run these MD workflows. Humans can do these cheminformatics tasks, but humans can't detect like contradictions in every single paper in literature against hundreds of papers. So ContraCrow combines claim extraction with PaperQA2 to detect contradictions in literature. So you input a paper and it chunks out. It splits into chunks, extracts claims and filters them, and then puts them into contradiction detection prompt in PaperQA2. And it gives you an answer, like a reasoning that you can read with sources, and then also a Likert scale of, yes, there's explicit contradictions to some literature, all the way down to 5, which is, I have no idea, I've never heard of this before, and 0 explicit agreement, like everything really agrees. So we took the LitQA data set that Andrew introduced and split half of them into true statements like the sky is blue, in half into not true statements like the sky is not blue, and put them all into ChemCrow, and we've got a AUC ROC of 0.842 as like a proof of concept that we're able to actually detect these in literature. So the next thing we did is we were like, okay, let's run it on 100 random papers. So we went in our database and found 100 random biology papers from all over the place, all sorts of journals from like 1982 to current, all over the place. And we found that all the way on the left here, most papers have pretty few contradictions that we found, which is great and expected. If we found like 15 on every paper, that would be insane. But if we look at the number of papers with each number of contradictions, we have 0, 1, 2, 3 being the highest, which is pretty reasonable. And we actually found 2.34 contradictions per paper on average in the papers we looked at. Then we use human experts, so the PhD students again, and ask them to first look at the claims that ChemCrow looked at and the reasoning and its score, and just, do you agree with it? Based on its reasoning, based on the literature it looked at, do you think this is a contradiction? And so for these scores of 8, 9, and 10, which we consider a contradiction if it's above that, humans agree with 70% of them, which might seem a little bit low to you. So then we asked humans, okay, you can look at every paper that, we're not going to ask you to look at every paper ever, but you'll look at every paper that PaperQA even considered and see if you think there's a contradiction. And so what we're looking at here is I sorted it so that it's pretty, but there's no like correlation along the x-axis. The stars are ChemCrow scores. So these are ones that it scored 8, 9, and then like 4 below. So agree, it all agrees, or it all contradicts. And like, you can kind of tell that humans don't really agree with ChemCrow all the time, right? Like all the way on the left, you see that it gave it a 9 and humans gave it like a 1. But if you look at the horizontal lines, we had like 3 humans on each claim. Humans don't even agree with each other on the large. Some of them literally gave like, yes, 100%, all of the literature agrees. And on the same claim, another human gave 100%, they all contradict it. So I think this shows that like humans in general agree with ChemCrow, and we're able to find contradictions in literature. And this is a really hard task to do. So this is something that we're using LLM agents to do that humans can't do. In fact, one of our contractors even said, this is the hardest task we asked them to do, and they don't want to do it anymore. So this is something that's a great example of how we can use these LLMs to scale into areas that are like not feasible or like good for humans to do. Mm-hmm.
I really like this this contradiction, because I feel like a lot of the papers I write are like almost like spite papers where I see at a conference someone claiming something, and then you go, and then someone disagrees, and you're like, I could prove that, right? Like I could prove they're wrong. And so I think it's really cool because like a lot of, science comes from contradictions. I was wondering if you've done any like structured searches like that. Like let's say, I'm in neuroscience. If I go to like Nature Reviews Neuroscience, and like for my topic, which is connectomics, I just download every review that mentions connectomics and put it in. Can you start getting like scientific concepts and then like testing them and have some metric of that?
Yeah, this is a great question. So this is something we've actually dove into this a lot actually for different topics. Originally, when we started this, we thought we would find really like hotly contested areas of science or like places to do research. A lot of what we found is like authors don't do their job finding literature and reading it before they write their papers. But certainly we also did look at topics, and the font is very small, I'm sorry, but we looked at across different journals. And so on the bottom, it's like kind of like topics. I don't have any plots of like the specific areas, but you can see like Nature Aging, Nature Biotech, Nature Chemistry. And this is not public data yet. We see that like Nature Aging, the number of contradictions per word is a lot higher than like Nature Methods, which is great because why would there be so many contradictions in Nature Methods? And then we asked an LLM to take the reasoning that the ChemCrow outputted and the claim, plus a little context that the claim came from, and whether it's a contradiction or not, and categorized it as inherited. So is this like introduction? Like it like cites a paper and the paper has been refuted, or it incorrectly cites it? Is this something that said that's like a speculation that was later disproven? Does it overstate the evidence? So if it finds like Pit bulls really like bananas, and it says all dogs like bananas. Very like absurd because it has that context. If it's just like an outdated claim, or if it's empirical disagreement, which is very common, right? Like papers can just find different things. Maybe they use different samples. Maybe they like just find different things. That's very common in science. And we find that like nature aging is really high, and that's because it's like you can kind of split it out and see the different ways and how. But we did look at different topics as well. Yeah. But then you have to download all the papers, which is the thing. Yeah.
Sorry, if you go back 2 slides for that benchmark AUC, it's 0.84. What is the contradiction struggling to detect, and how are you guys addressing that?
Yeah, so this was, I don't remember what the recall of like QA was at this point, but it was also not 100. Do you remember? Yeah, so the recall of lit QA, meaning like, can it, if given the question, did it find the right paper in general? Because there's only one paper, was like 67%. And so we're getting 53%. So it's possible that it's literally just not finding the paper in most cases. I didn't dive really into farther into that because this was kind of just like a proof of concept that it's able to detect them. Yeah. But the recall of these papers in general was not 100. So it never stood a chance there either. Any other questions? Okay. Yeah. I think that's all I mostly have. Just final conclusions. We kind of, with ChemCrow, we found like, okay, cool, these LLMs can do science, but they're limited by the tools. With MDCrow, we found we can do more complex tasks, but it tends to kind of struggle, and open source models are kind of equal still to the frontier models. And then with ContraCrow, we found, hey, this is a really hard task, but it's necessary for science. And if we use ContraCrow, humans agree with 70%, which is a pretty good amount. And then also preprinted papers, shocker, have more contradictions than most peer-reviewed journal papers. So that's kind of the, and then obviously we can scale much easier with ContraCrow.